Hair is a filamentous outgrowth of protein possessed by all mammals. The hair of non-human mammals is commonly referred to as fur. Depending on the stage of life, hair may or may not be visible. Although there are limited characteristics to determine a hair's specific origin, human hair has a collection of traits that make it distinguishable from other animal species. In a forensic context, hair examination evidence is typically used in conjunction with other evidence because of its low discriminating power and circumstantial nature. The purpose of hair examinations is to find possible associations between people, places, and objects. Hair grows in a structure called the hair follicle. It can be divided into 3 different regions: the root, which is termed the proximal end, the shaft, and the tip, commonly referred to as the distal end. There are several macroscopic characteristics that can be observed in hair with the naked eye and with a stereo microscope under relatively low magnification. Color, shade, and intensity can be noted. The shaft form is also important. It can be straight, curled, waved, kinked, or curved, for example. The thickness of the hair strand can be measured or described as fine, medium, coarse, or varied. The length of the shaft is also a significant feature. The 3 morphological sections of hair that exhibit important microscopic characteristics are the cuticle, the cortex, and the medulla. The cuticle is an outer layer of dead cells, or scales, that cover the shaft. The cortex is the unorganized protein matrix that surrounds the medulla. The medulla is a canal that houses foreign debris, medications, metals, and other substances that the body secretes. These regions can be examined under compound light microscopes with moderate to high magnification. The cuticle scales always point to the distal end of the hair. They form into 3 different pattern categories. The coronal structure is a crown-like pattern that resembles stacked cups and is common in thin hairs. Spinous or petal-like cuticle patterns are triangular in shape. The imbricate or flattened cuticle structure involves many overlapping scales that are wide and short. The cortex contains many microscopic characteristics found in hair. The distribution of the pigment in hair is an important consideration. Small granules of pigment may be visualized, and their color and distribution along the length of the shaft will vary. Large aggregates of pigment, called ovoid bodies, may also be present in the cortex. They typically occur in regular intervals along the shaft. Other microscopic objects that may appear in the cortex are small fusiform bodies filled with air called cortical fusi. These are larger than pigment granules, and their sizes, locations, and frequencies are important to observe. The medulla can appear in many forms depending on the species of hair. Several patterns can be observed, including ladder, lattice, vacuolated, and amorphous forms. It can be continuous, discontinuous, or even absent. The width of the medulla also varies. The medullary index, or the ratio between medulla diameter and the total diameter, can be an important characteristic for determining species. Human hairs generally share the same microscopic characteristics that make them distinguishable from animal hairs. The cuticle of human hair always has an imbricate pattern. Animal hair may exhibit this pattern, but can also exhibit the other 2 types. For example, coronal patterns are often seen in rodents, and spinous are often seen in cat and seal hairs. The pigment distribution in human hair is typically spread throughout the length of the shaft. It is also generally distributed evenly or slightly towards the cuticle. This is different than an animal hair, which can have banding, or a pattern of color change across short distances of the shaft. This pattern is typically seen in guard hair, which is the outer coat of an animal that provides protection. Animal hair pigment also tends to distribute closer to the cuticle. The medulla of the human hair, when present, is amorphous and smaller than 1/3 of the overall diameter of the hair. The medullas in the hair of many animal species are relatively large and form distinct patterns, like in cat hair. The root of human hair is commonly shaped like a club, whereas animal hairs have root shapes that are highly variable. There are a limited number of conclusions that an examiner can reach when analyzing hair samples. Statistics and probability are not involved in the formation of a conclusion. Interpretations are made from the examiner's observations alone. One conclusion that an examiner can reach is the identification of the hair as human or non-human. When comparing known and questioned hair samples, three possible conclusions can be reached. The hairs can be termed similar, dissimilar, or inconclusive. These are based off of the microscopic and macroscopic characteristics observed. The degree of dissimilarity can vary depending on the amount of gross differences between samples and the amount of intra-sample variation. An examiner cannot confidently state that the two hairs came from the same source, so the significance of the conclusion is restricted. Therefore, it is commonly used in conjunction with other evidence in court.